Hello and welcome back. Today we continue our journey into macromolecules and we start a little side quest here on proteins. We'll have two different sets of notes on proteins. This is obviously the first one, and the next one will be specifically about enzymes. So let's just dive right into proteins and get started on that. So while we begin, let's just go over some fast facts about what are proteins. So the monomer of a protein is an amino acid. Amino acids are like so important to everything. They are what gives humans their traits, gives everybody their traits, right? Because DNA, as it goes through protein synthesis, codes for different proteins, which allows us to show what the proteins are coding for. So there are 20 different amino acids coded for in the genetic code. So different combinations of those 20 amino acids gives all of the variation that you see in all of the species that you see. The order of amino acids will tell you how the protein will fold, and based off that will tell you how the trait will look. Amino acids are bonded by peptide bonds. We talked about this in an earlier set of notes. The protein shape gives us its function. And the R group of an amino acid is what makes it unique. That's what changes between the 20 different amino acids. All amino acids are shaped like this. You have an amino group, you have a carboxylic acid group, you have a singular hydrogen, and then you have an R group. And the R group is what changes. So all of this on the bottom here will be the same for every single amino acid, so you should know this structure, that the R group will be different as we learn about different amino acids. I mentioned protein folding, which will eventually show you what the trait is looking like. So a primary structure is just amino acids matched up to each other. So multiple amino acids just in a line, basically. Secondary structure is when you start to connect those into sheets, still relatively two-dimensional, just sort of making sheets. Tertiary structure is your next one. That's when you really start to fold and you're starting to see this 3D structure of a protein. And then quaternary structure, that's your big one. That's like where you're getting all these proteins that actually have like real jobs that we care about. And these quaternary structures are multiple tertiary structures bonded together. Like hemoglobin, for example, is a quaternary protein. Has a really important job because it can handle that load of instructions. So that is how proteins go from a singular amino acid all the way up to a working functional protein. The proteome is a word that maybe you're familiar with or maybe you've never heard of, but a proteome is similar to a genome. A genome is, all of the genes and the DNA within a human being or any species really, but I'm a human, so I tend to strictly think in terms of human beings. And a proteome is the same kind of thought process, but it's just a list of all the proteins produced in a cell, tissue, organism, whatever it may be. But it's all the proteins that are produced. Each individual will have a different proteome strictly because there are no two individuals that are exactly identical. I know you're thinking, what about identical twins? Well, the methylation of their genome is different, which produces different proteins, so slightly different. Very similar, but slightly different. And if you're like, wait, what's methylation? We'll get to that, don't worry. The functions of proteins, you've probably heard of these before. Structural, like muscle contraction, right? Like most people think of muscles when they think of proteins. Tensile strengthening, so you have anything to do with muscles is kind of thanks to proteins. Transport moves molecules like hemoglobin, I mentioned that earlier, carries oxygen to your cells. Movement, like cilia and flagella, move cells. Defense, when we're talking like antibodies to protect ourselves against infections. Hormones, usually triggered by the brain to monitor body functions. Insulin, FSH, follicle stimulating hormone, and LH, luteinizing hormone. All three of those are proteins. And membrane-bound proteins found in the cell membrane that helps with transport in and out of the cell, have binding sites for other proteins. to bind to the cell to allow for additional whatever function they may be to occur within that cell. And then maybe one that you're most familiar with, cell regulation, so controlling cellular functions, aka enzymes. Enzymes are a special type of protein that catalyze or speed up chemical reactions. Our entire next set of notes is about enzymes, so get ready. They can be denatured by heat, altered by the pH of whatever environment it's in, so heat and pH are like its nemesis, but ideally they will work in their perfect conditions so that they can continue to speed up chemical reactions. Now, this is a big slide. I would suggest pausing, writing it down, because this is all of the proteins that you are required to know specifically about. So there are 6 of them. You are actually required to know these 6 specifically and what they do. So going through them quickly, rubisco is found in plants. It helps with photosynthesis. Insulin is produced by the pancreas, helps monitor blood sugar levels. Immunoglobulin is an antibody that recognizes an antigen, helps with the immune response. Rhodopsin is found in the retina of the eye, useful when you're in really like dim lighting. Collagen is connective tissue. It's really found in skin, tendons, and ligaments. It's like if you like feel the back of your ankle, that tendon, right? Your like Achilles heel, that is made up of collagen. And then spider silk. I know, it's like, this is weird. Spider silk, yes. I have seen so many questions about spider silk. It's a protein that's spun by spiders for making webs. I'm sure you could have figured that out on your own. Has tons of uses, but it is a fibrous protein, and that's the key there, that spider silk is a protein. Now, IB is very particular about what you need to know, and this is one of those very particular things that you need to know. Sickle cell anemia. Now, if you took biology, you know, we've talked about sickle cell anemia before, but basically what it is a genetic disorder that changes the shape of your red blood cells. So these donut-looking things, these are your normal red blood cells, and these kind of crescent moon-shaped red blood cells are sickle cells, and sickle cells are not able to carry oxygen as well as your normal red blood cells, and they also hook into each other, so clots are much more frequent when sickle cells flow through the body. So what causes sickle cell? It is a single amino acid chain in hemoglobin. One single amino acid changes the entire shape and functionality of your red blood cells. I think that's absolutely crazy. But the single change is, instead of your typical glutamic acid, it now codes for valine. And so that one change changes all of this. So you will need to know that that is the change that causes sickle cell anemia. We'll talk more about sickle cell anemia just like on its own, but I wanted you to be aware that that was what was responsible for the change. And then last but not least, we're going to talk about amino acid structures. I am not going to have you memorize all of the amino acid structures, but I would like to talk about them so that you can kind of see, especially methionine, this big one right here on the right-hand side. This methionine is probably going to be the only one that you know everything about. Methionine is the start codon in mRNA. It is always the start codon in mRNA. There is never, ever, ever a different start codon in mRNA. It is the first amino acid in all polypeptide chains and first produced in transcription. It is always the beginning. So it has a pretty large R group, as you can see. You notice that the bottom is the same, the amine group is the same, the carboxylic acid group is the same, the singular H is the same, but the R group is different, okay? So you should be able to identify methionine strictly based on its R group. Now, this is a pretty complicated R group, but the simplest amino acid, Look at little baby glycine. It only has a hydrogen in the R group. So there's like nothing. It's so simple. So glycine is the simplest. Methionine is not even the most complex, but it is the most used, I would say, in terms of us talking about it. Now, these are the rest of the 20 amino acids. Again, I am not requiring you to memorize the R groups of them, but you should be able to identify methionine, and you should be able to know that valine is the change in sickle cell anemia. But I did want to just point out that all of these like colored boxes are the different R groups of the different amino acids, and you can see that some of them are quite complicated and some of them are really not. So there are multiple differences in the R groups. No two are the same, of course, that's what makes them different, but it's interesting to look at them all. We will be using all of these, but you will not have to memorize the R groups, but you should be comfortable in the idea that there are 20 and there are some that we are going to focus on more than others. All right, great job, and I'll see you next time.